The second line of defense is more sophisticated than the first line of defense because the immune system should actually do something to deal with the pathogens that have penetrated the first line of defense. You want to imagine the second line of defense as being a combination of the FBI and the armed forces involved in combat or rescue. We're going to classify the second line of defense into three types: protective processes, protective proteins, and protective cells. So we'll start with the protective processes. There are two types of protective processes that we will look at. One of them is inflammation, and the second is fever. Both inflammation and fever are beneficial to the body unless they go out of control. The second is protective proteins. Let's look at two types of protective proteins. One of them is the interferon, which is an antiviral protein, which we will look at closely in a bit. The second is a complement protein. Complement proteins are a set of proteins floating around in the blood, waiting to be activated by the presence of a pathogen. The third group is the protective cells. We will take a look at two major protective cells at the second line of defense, and they are the natural killer cells and the phagocytes. The natural killer cell is a kind of lymphocyte, and the phagocyte is the macrophage or monocyte. Let's take a look at the protective protein called interferon. Interferon interferes with viral replication. In order to understand how interferon functions, we need to understand how viruses operate. A virus cannot multiply by itself. It must enter your cell and then use the resources in your cell in order to multiply. Once the virus multiplies, it then lyses the cell, the host cell, basically destroys it, and all the viruses infect the neighboring cells. This is how they operate. The interferon stops this from happening. So let's see how. We have two cells here. The cell on the left is being infected by a virus. The virus is shown in yellow. The virus does not enter the cell, but it introduces its genetic material. The genetic material could be either DNA or RNA. Once the genetic material enters, the cell responds by becoming activated and making a protein called interferon. Interferon is shown in pink. Once the interferon is made, it can now leave the cell via exocytosis and enter the neighboring cells. So interferon leaves the cell, reaches neighboring cells, and influences the neighboring cells and asks them to prepare for viral infection. So here's your uninfected cell. This cell does not know that there is a virus in the vicinity. Once the interferon signals this cell, the uninfected cell makes antiviral proteins called AVP. These antiviral proteins are like little weapons that are ready for a viral invasion. When the virus actually arrives and introduces its DNA or RNA, AVP blocks viral replication. If viruses cannot multiply, they cannot survive. So the AVP blocks the viral replication and basically stops viral infection. So let's recap. Interferon is a protein that is produced by an infected cell, and its job is to signal healthy cells in the neighborhood to prepare for viral invasion. The last component of the second line of defense are protective proteins called complement proteins. As their name indicates, complement proteins enhance or complete the action of other proteins and cells of the immune system. There are several kinds of complement proteins classified into many groups and subgroups. But what we're going to look at is how complement proteins in general influence or enhance immune functions. So let's take a look at the first function. Complement proteins help to enhance inflammation. As we know, inflammation is made possible by mast cells that release histamine and make capillaries leaky, and they also depend on neutrophils and macrophages that kill the bacteria. So the complement proteins stimulate the mast cells and intensify the action of neutrophils and macrophages. Together, they enhance the process of inflammation. The next function of complement proteins is to make pathogens more appetizing. Yes, you heard that right. The complement proteins act like curry powder on your food. Here's the pathogen shown in blue. The complement proteins shown in pink, they coat the pathogen as if you were to season your food so that they would be more appetizing to the phagocytes. This actually has a name. We call this process opsonization, which means make the pathogen into food. And this really helps because the phagocytosis is intensified. The next function of the complement proteins is to make holes in the pathogen or any enemy cell. It is not unlike what happens with the perforins. Here, the complement proteins make multiple holes on the enemy cell, causing the enemy cells to be destroyed. We call this cytolysis. Cytos means cell, lysis means breakdown.